afternoon, everybody. My name's Fiona Hunter, and along with my colleague Emma Manners, I'd like to present to you today a guide to exploring drug-like compounds and their biological targets in ChEMBL. So, first of all, what is ChEMBL? ChEMBL is a database of activity data that links compounds to their biological targets. And what is ChEMBL used for? So it's used for a variety of different purposes for drug discovery, to provide insights, to create tools, and just as a general resource for information. So if we start on the top left-hand side here, then the data in ChEMBL is used to feed into other platforms. In this example, this is the Open Targets platform, which aims to prioritize targets. So the information on marketed drugs and drugs in the clinical development pipeline that link to their target via their mechanism of action feed into the Open Targets platform, so you can try and prioritize targets should that be of use to you in your research. If we look at the central panel here, then information in ChEMBL contains information on chemical probes. So these are high quality chemical tool compounds that are used for preclinical drug discovery. And we're involved as part of the ChEMBL group with projects such as EUB Open that aim to collate information on chemical probes. Data from ChEMBL is used for drug repurposing. On the top right-hand side there, then the data, if you're trying to find out which targets are druggable, for example, which human genes do not yet have drugs that interact with them but might be possible for treating a particular disease, then you may be able to repurpose existing drugs that are used for other drug indications. So data from ChEMBL is used for that purpose. If we follow down to the bottom right, then there's a screenshot of the front page of a Nature article from a couple of years ago, where the data in ChEMBL was used to find information on existing drugs for proteins that could be used for targeting neglected tropical diseases. In this case, schistosomiasis. In the center bottom, then we have a tool which allows molecular prediction. In this case, the data in ChEMBL was used to provide a tool for malaria inhibition prediction. So if you have a novel chemical compound, you could submit it to this in silico tool, and that will identify whether it may have anti-malarial properties. And finally, the final example on the bottom left, the data in ChEMBL is used for druggability analysis. We have a screenshot of an Illumina array, which is used to find genes that encode proteins that are known efficacy targets of small molecule and biotherapeutic drugs. So it's used for screening for druggability. Right, now moving on. So a little bit of a summary about the database content of ChEMBL. So the first thing to say is that ChEMBL is a very large database of bioactivity data. The data is open access data, so it's freely accessible over the web. Anyone can use it regardless of the purpose, whether they're academia or pharmaceutical companies or small companies. The core of the database is primary medicinal chemistry literature covering nearly 40 years of research. So in the top left here, we have scientific literature, which we manually extract, and we include that within the ChEMBL database. We also have deposited data sets. For example, there are data sets that look at neglected tropical diseases and data sets that come from pharmaceutical companies that are included within ChEMBL. We take other data from other relevant data from other public databases, such as PubChem. We extract their confirmatory assays. If there's information in patents, then we may also incorporate that within ChEMBL. In particular, we've been involved with a project called IDG, Illuminating the Druggable Genome, which aims to focus underexplored targets. And then we look at the full scope of the drug discovery pipeline, which includes toxicity. So we have toxicity reference data sets such as TG Gates and Drug Matrix, and also other information on drugs. So by drugs in this case, I mean drugs both that are on the market and those progressing through the phases 1, 2, and 3 of the clinical development pipeline. And we have information on that. So in all, we have around 2 million compounds in the current version of ChEMBL. So how do we extract the data? So if we take a scientific article from the published scientific medicinal chemistry literature, then we would manually extract the information in the center panel there. Then we have a description in blue of the chemical compound that's being described within this article. We extract the structure and the information around the compound and incorporate that within ChEMBL. And then equally on the left-hand side, we have the target, which the compound is interacting with. In this case, it's a thrombin. So we would extract all the information around the target. If that target's a protein, for example, we would register it and assign it a UniProt identifier if that's available. And then the information that links the compound to its target is the activity, or sometimes called the bioactivity. So that might be measured in this example. We have a Ki measured in nanomolar, or a APTT, which is a measure of blood clot. So what's important to say is that all the bioactivity data from each paper is manually curated and incorporated within ChEMBL. So the scope of ChEMBL includes compound binding to targets, such as in this example, but also functional assays, in vivo efficacy assays for whole animals, in vitro and in vivo ADME and pharmacokinetic data, and toxicology measurements. Now, I'm going to provide a little bit more detail about the concept we use for targets within ChEMBL. If a compound binds to a single protein, for example, in the bottom left there, we have PDE5, so phosphodiesterase 5A, then this is a fairly obvious example to measure and understand. But it can be more complex than this. For example, on the bottom right, near the bottom right, we have a protein complex group, which is a GABA-A receptor, which is composed of various different subunits, alpha-1, gamma-2, beta-1. And a compound may bind to only one subunit of the protein complex. So in this case, we would register a protein complex as the target within that assay. But equally, particularly in some of the older journal articles, then we don't necessarily know which protein within a group has been measured. For example, if you have a protein family and all the paper says is that it binds to a muscarinic receptor, then we would annotate a muscarinic receptor protein family as the target within ChEMBL. Now that's detail about proteins. ChEMBL also considers other targets, so nucleic acids, but equally non-molecular targets. So if a target could be a cell line or a tissue or a subcellular fraction or indeed a whole organism, and each of those would be recorded as the target within ChEMBL. So this is a summary that can be a useful reminder when looking at data on the web interface. So when we type into the search box on the web interface, in this case, in the screenshot, there's an example given for dopamine, then it returns a number for each different category of ChEMBL entity for compounds, targets, assays, documents, cells, and tissues. So compounds and targets are fairly self-explanatory. So compounds, the chemical structure and the properties on which the experimental data has been measured, and the target is the biological system. The assay is the description of the specific experiment, and then the document is where the data has been reported. So that might be a scientific paper. But equally, if it's applicable, then we would, you could think of the entities as if the experiment has been analysed within a cell or within a tissue, then we would record that type of chemical entity. So you could, and the cells and the tissues are mapped to specific ontologies. So if you want, you could start from a particular cell line or a particular tissue type and find all information that has been recorded for that cell line or that tissue type. So when you view the bioactivity data within ChEMBL, then you come back with a screen that looks a little bit like this, a table, if you like. And working from left to right here, we have the first couple of columns on the left relate to information about the compound. Then the next few columns are to do with the activity, the bioactivity that links that compound to a target. Then the third column, we have the assay. a description of the assay, a mapping to any ontologies, in this case, the biontology. Then we have information about the target name, which organism it's been recorded in and what type, whether it's a cell line or a protein, and then information about the source. And on the bottom here, I've included a note to say ChEMBL. So one of the things that can be difficult within bioactivity data, for example, if you're interested in a series of compounds recorded against a particular target, particularly a protein target, they often have had activities measured, but in different units and using different methods. So you might have an IC50 value or a KI or a KD, and these all have different units and therefore are difficult to compare. So as a result, ChEMBL has developed a concept called ChEMBL, which is the negative log 10 of either of any of these bioactivity measurements. And this provides a quasi-equivalent bioactivity measure, so you can compare different data types in a relatively straightforward manner, although they're nearly equivalent, although not entirely equivalent. So that can be a helpful means of accessing some of the data. So all the information I've presented on the last few slides is to do with compounds binding to targets. Now I have one slide to do with the drug information in ChEMBL. So ChEMBL does a lot of work to annotate information for drugs and for clinical candidates. That's drugs which are progressing through the three different phases of clinical development before they are approved and put on the market, at which case we call them a phase 4. So ChEMBL manually annotates any mechanism of action that links the target to a drug. Now that differs from whether you have a compound that has an activity. So if you have a compound that has a bioactivity measured against a specific target, then it would be given IC50. But in this case, we're saying that we believe that this drug has a mechanism of action to treat a specific disease because we understand from other evidence that it works at this target. So we've put a lot of information into annotating this type of information, mechanism of action, but also drug indications. So which disease has the drug been used to treat, and at which stage of the clinical development pipeline is it currently available for? Is it on the market or still progressing through different clinical phases? So this information does not come from the published scientific literature, but from other information sources, including clinical trials and the medicinal product labels for marketed drugs. And on the right-hand side there's a screenshot of the report card that you would find for a drug that's used to treat diabetes. Note in the middle there's a series of icons that provide a quick means of assessing what we know about that drug, whether it has, how it's administered, whether it's a small molecule, whether it's chiral, whether it has a black box warning, and so forth. Right, so if we've got all this information, great, but actually what's useful, and the first place that most people would come to, is to look at the ChEMBL web interface. So this is a brief tour of the landing page, the home page, if you like, for the ChEMBL web interface. So typically on the top right-hand corner, you would input a keyword that you're searching for, perhaps the name of a target like CCR5 for chemokine receptor type 5 or something different. And that will pull back the different ChEMBL entities that I talked about, how many compounds, targets, assays, and documents are available that can be searched for with that keyword. Now, if you want to return to the main ChEMBL landing page, then we go to the top left. And if you want to access different types of data, then in the center of the image, there's what we call a bubble plot. So you can click on these individual bubbles and that will take you through. The largest one there is for the 2 million compounds. You'll access the table that I showed you earlier for the compounds, but equally you could access assays or targets, for example. However, if you want to find the drug information, then that's a good place to start. There's a bubble for drugs and their mechanisms and indications. And I'll just mention in passing, there are various other data views. So if you click on the small arrow to the right, you'll see other data views, for example, for the protein target tree, and it's worth exploring that when you look at the ChEMBL database. Right, so that's a brief, so now when we start to drill down through the ChEMBL web interface, this is a type of information that you might see if you search for a particular target, in this case a cannabinoid CB1 receptor. So it returns 8 different targets which we've clicked on, and there's a series of columns. It's laid out in table form. So you can select the data using a checkbox on the left-hand side. And then in the top, you can view the activity data further, should you wish to do that. Equally, if you want to know more about that specific target, you could click on the target, and that will take you through to the target report card. You can download the data in the top right, or you can filter on the left-hand side by various different filters which are tailored depending on which entity you're in. And equally, you can view it as a heat map should you prefer that to a table. So how do we access this data? Well, I've told you and given you some examples as an overview of accessing the website, but equally in the bottom left corner of this screenshot, then you can download the whole database as an SQL format in various different flavors. You can access the information through the web services should you wish to incorporate programmatically the data into your workflows. We have a number of different API endpoints to do that. And equally, you can access it via the semantic web. So that's a brief guide to compounds and their biological targets in ChEMBL. Now I have a couple of slides providing information on our sister resources that can also be used for your research. And then I'll hand over to my colleague Emma, who will go through a couple of examples. So this is the first of our sister resources called ChEMBL. Now ChEMBL is a patent database for chemical data. And why this is useful, the information often comes into the patent space if you're interested in a particular compound. The information is often available in the patent space before it would be published within the scientific literature and therefore available via ChEMBL. So ChEMBL provides search access to all patents, not just chemically annotated ones. In total, there's about 140 million patents within this database. And as I'm sure you can understand, because of the scale of this, 140 million patents, but each patent can run to sometimes tens or even dozens of pages, then we have a fully automated pipeline for extraction. So there is no manual curation within ChEMBL. ChEMBL extracts chemical structures from different sections of patents, from their titles, descriptions, abstracts, and claims, and it takes it from US patents, from European patents, from Japanese patents, where there are English abstracts, and from world patents. So if you can't find information on your particular compound, then please come and look at the ChEMBL resource. What's very exciting, there's been a lot of work going on behind the scenes for ChEMBL, and coming soon we have an API endpoint that will be available and also a new web interface. Now moving on to our second sister resource, this is UniChem, which is a compound mapping resource between different resources. So UniChem maps compound identifiers and their chemical structures between different resources in a very efficient manner. So it uses InchiKey to map. So in the center of the green cylinder there, then you can see different identifiers for different resources. An example is given for ChEMBL 741 with its InchiKey, and we can find the same chemical structure in each of, in this case, 5 different other resources in ChEMBL, Drug Bank, FDA, PubChem, links, etc. So it's, I should say that InchiKey is a representation of the chemical structure, and this is what's used to do the mapping. So it allows you to take one resource to start with and map to 40 other resources as your output. So this is accessible again via web interface, via download, but also what's very exciting, a lot of work's been going on behind the scenes, and we have a new website coming soon, so please keep your eyes peeled for that. Right, now I'll pass you on to my colleague Emma, who's going to take you through an example to explore drug-target disease relationships.
Okay, so now that we have an overview of the ChEMBL database, let's explore the kind of questions that we can begin to answer using ChEMBL. So let's take dengue fever as an example, as this illustrates the type of information that can be found within the database. So dengue fever is a viral infection that is spread by mosquitoes. There is no specific treatment or cure, and instead the symptoms are managed. A vaccine was FDA approved in 2019, but with restricted use. If we wanted to develop a drug for dengue fever, how could we use ChEMBL to explore some of the existing data? For example, we might want to know, are there any approved drugs or drugs in clinical trials for dengue? What compounds are active against dengue? And are there any active compounds with drug-like properties? Next slide. So, to explore drugs and clinical candidates for dengue, first, click on the indications view, which is found on the ChEMBL homepage, and then search for dengue using the free text search bar. Next slide. Compounds that have been tested in clinical trials for dengue fever are then displayed. The highest phase a compound has reached is phase 3. We can see here that compounds such as ivermectin have both the indication dengue and severe dengue. However, if we click on Browse Drugs, then we can explore the set of distinct drugs. After browsing drugs, we can then see that there are 13 distinct drugs that have been tested in clinical trials for dengue fever, and these can be found within ChEMBL. So as mentioned earlier, ChEMBL contains a range of compound types. So as a result, our set of drugs includes vaccines, an antibody, and a suite of small molecules, all of which have been investigated in clinical trials for activity against dengue at some point. Within the compound set, we can also see approved drugs. In these cases, the max phase is equal to 4. However, these drugs are not approved for dengue, but for other indications. So approved drugs can be useful as potential candidates for drug repurposing. So as mentioned earlier, drug repurposing is a strategy that explores new uses for existing drugs. It can have advantages over the development of an entirely new compound. We can look more closely at some of the data by clicking on individual compounds and reviewing their compound report cards. By doing this, we can see that most compounds in our list are at phase 1 in the clinical trials for dengue. Valipirivir has also been explored for efficacy against hepatitis C, another viral infection. We can also see that chloroquine is approved for malaria. So overall, by using ChEMBL data, we can gain an overview of compounds tested against dengue that have progressed to clinical trials at some point. But what if we're interested in all compounds that have been tested for potential in dengue, and not just those that have progressed to clinical trials? Well, it's also possible to view all ChEMBL compounds that have been tested against dengue-related targets. So to do this, you can first search for dengue using the main search bar. Secondly, you can click on the Targets tab to view the set of targets. The first point to note here is that our targets vary. So we can see whole organisms from phenotypic screens, as shown here by the different strains of dengue. Our targets also include single proteins. So in this case, you can see the dengue non-structural protein 3. So alongside each target is a graphical display illustrating the number of compounds that have been tested against the target, as well as the number of bioactivity data points. We can use these histograms to quickly view bioactivity data or compounds tested against a single target. Alternatively, we can click Browse Activities to return a summary table of all the bioactivity data across all 6 dengue targets, as we'll see in the next slide. After browsing activities, our summary table includes over 4,000 bioactivity data points across a range of assays, all tested against dengue-related targets. So this provides a good starting point to explore drug-target relationships. So importantly, references are available for all data points and can be checked for compounds of interest. These are shown in our table as document IDs. So now that we have our bioactivity data, we might be interested in filtering the data to focus on compounds with the most potential. For example, those that are considered to be drug-like. So options for this include filtering the data for potent compounds, for example, those with a ChEMBL value greater than or equal to 6. We can also filter for compounds that comply with the rule of 5, an indicator of drug-likeness. We can apply these filters using the left-hand filter panel. We can view or add additional filters by clicking on the filter settings icon, as shown here, or we can add additional columns to our table by clicking on the Show Hide Columns button. Here, I've added the rule of 5 column, which is not shown by default. So, after filtering for potency and rule of 5 compliant compounds, we have a subset of 159 activities. So to explore these selected compounds in more detail, we can click Browse Compounds. So at this point, we've now extracted a set of potentially potent drug-like compounds that display activity against dengue targets. Next, we can review additional information on our selected compound set. For example, we might be interested in reviewing any existing toxicity or selectivity data. So here, clicking on the heat map icon will provide a visualization of compound target pair data. So in this slide, a subset of the heat map data is shown for clarity. In this heat map, colored squares indicate that bioactivity data is available. Darker blue indicates a higher ChEMBL value for a compound target pair. Within our results, we can see an approved drug, dasatinib. Approved drugs can be useful in some cases as they may have more associated data and therefore provide a good starting point. A number of our compounds have been tested against several dengue strains, which can provide an insight into their activity across related targets. You can see these targets by looking at the top of the heat map. In addition, activity against other non-dengue viral targets may be available. And we can see here that some of our compounds have been tested against other viral targets, such as hepatitis C, indicating there may be some potential for broad antiviral activity. We can also explore activity against protein targets, in addition to our strains. So, for example, cytochrome P450s. Importantly, cytochrome P450s are relevant to in vivo metabolism. Finally, cell permeability may be important, especially for activity against virus-infected cells. So in the heat map, we can see that the CACO2 cell line is shown among other targets. This cell line is often used to assess cell permeability. So overall, the heat map data summary helps us to evaluate ADMET data that may be relevant to drug development and also to review the selectivity of our compounds, which may provide an insight into potential off-target effects. All data points within the heat map can be investigated in more detail by clicking on the corresponding squares. So hopefully this has provided a brief overview of the type of data that can be found in ChEMBL and the type of questions that we can ask. But there are numerous applications. So for example, we may want to review similar compound sets as well as structure and reactivity data. We may be interested in structural biology applications, such as review of bioactivity data linked to crystal structures. Within ChEMBL, we have information on black box warnings and withdrawn drugs, and that can be useful in toxicity analyses. And finally, a carefully selected and checked dataset can also be a useful starting point for modeling applications, such as target predictions. And notably, ChEMBL contains negative data where available, which may be equally important for your research question. So we have some additional resources, webinars, and documentation that may be useful, and you can find the links here in this slide. But if you have any questions at all about using ChEMBL, then please get in touch with us on the help desk. We welcome any queries, feedback, and suggestions. So finally, we'd like to acknowledge the current chemogenomics team, as well as those who have previously been involved in the development and maintenance of the databases. Thank you.
All right, so with that, I'll hand over to Emma and Fiona for Q&A.
So shall I start with the top question on the list here? If I read it out, and then we can think about how we should answer it. Brilliant. So the first question says, currently in my project, I have identified a single protein that can be used as a new antifungal target. How can I use this website server? Should I load the protein sequence of my single protein or writing of the protein name and description is enough to find a drug candidate. So you could do, yeah, Emma, would you like to go ahead?
Shall I start?
Okay.
So actually you can use both approaches. So you can use your protein name and type that into the search bar and then review the targets and see if there are any compounds that have been tested against that target. We also do have the option for a sequence search. So if you go to the advanced search options on the web interface, you can type in your sequence and then look for anything similar in the database, and again, review all compounds that have been tested against those targets. So there is an option to do both. Fiona, do you have anything to add?
No, I was going to say exactly the same. You can do both approaches. So shall I read out the second question?
Yes.
If a molecule has no targets, is it possible that that molecule does not target something in particular, or a target has not yet been identified? So I should start. I'll attempt to answer this, and then Emma can add to anything if she sees it, if she thinks it necessary. So typically we wouldn't include data within ChEMBL if the molecule can't be associated with a target because there's no bioactivity data. However, the caveat to that is within ChEMBL we have a concept of compound families where we have one parent, which is the same as the salts but without the counter ion, so it's a salt stripped parent. And there are some cases where, for example, we'll have a compound which is the salt, which we have information that links that to a target. So we've got bioactivity information on the child form, the salt form, but we don't have any information on the parent form. And in that case, the parent molecule may not directly link to the target, only through the salt. So it says, is it possible that that molecule does not target something in particular, or a target has not been identified yet? So the other side of that is when we annotate the information to do with drugs. That's known drugs that are on the market, or those in the phases 1, 2, and 3 of the clinical development pipeline. So in this case, we may know, because we have a known mechanism of action, that we can annotate the target to the drug. So we should be clear about the distinction we're talking about between a compound and a drug or a clinical candidate. So that, or in some cases, we may not yet know that mechanism of action to link the drug to its target, or the information may be known but just not yet curated within ChEMBL, so not annotated in ChEMBL. Is there anything that would be useful to add to that, Emma?
No, I agree with you, Fiona.
Okay. So should we move on to the next one? Can I use those tools for natural product studies or metabolomics analysis? Emma, would you like to start with that one?
So I'll start. So yes, so we do actually curate some of our compounds and flag whether we believe they're natural products. So you can go through the database and select and filter for those compounds, and then look at the activity data associated with those. Again, you can use any of the data within the database for your studies and research. And as for metabolomics analyses, there are other databases that we can that are integrated with ours. We have cross-references for our compounds and for our targets as well. So it may be possible that you can then review those and then link the data together. But yes, there are various ways in which you can use ChEMBL for these applications. Fiona, do you have anything to add?
No, that was exactly my comment, that there is another EBI resource that looks at metabolomics, but we also incorporate relevant data from them to ChEMBL. Right, the next question. How can we use your database and web server to know how the drug option is competitive and its affinity to my target? How can I know if it binds more to my molecule than the present cellular proteins that interact with my protein of interest? So I think some of the heat maps that Emma presented would be useful for that, where you can see the compound, assuming that your compound is available within ChEMBL, then you can see a compound binding to various different protein targets or other targets, and you know what their affinity is. But equally, if there isn't your exact compound within ChEMBL, then you could find a similar compound, and there's, within the advanced search options, you can search for, you know, say a 95, 99% chemical similarity to your compound, and then look for targets that interact with that, if that's relevant. Emma, would you like to add to that?
No, that's absolutely fine.
Okay, the next one. Good afternoon, thanks very much. How can you use DNA sequence of a gene to find a drug target? I think that means refine a drug target.
Okay, shall I start with that?
Yes, on you go.
So you can't actually use a DNA sequence on the ChEMBL interface, but if you can convert that DNA sequence to a protein sequence, for example, taking the protein sequence from a site like Uniprot, then you can use that to perform a sequence similarity search through the interface. So using the advanced search options, you can put your protein sequence into the interface, and then you can get a list of similar proteins, and then you can find the associated bioactivity data. It's also possible, if you have a protein accession, you can use that directly in the search bar. So I hope that answers your question. Fiona, do you have anything to add?
No, that sounds quite comprehensive. So the next question, I have seen the same compound listed under several IDs on ChEMBL. Is this intentional or a result of human error? I think in this case, the best thing is to know exactly which identifiers that you've seen that are the same, and we can have a look into that. So anything that you think might be an error, human error or other error, then please just email us on the ChEMBL-help email address, and we'll endeavour to help you. We're always very keen to accurately curate as much data as we can, and the only way we can do that, we can't spot all errors ourselves, and clearly there will be errors in a database of this scale. So we're always very open and hopeful that if you pick up on something, then please get back to us. Emma, did you have any comments?
No, I agree. So, I mean, occasionally you can see similar compounds that differ slightly in their stereochemistry, or it may be, as Fiona mentioned earlier, that we've got alternative forms. So one compound may be a hydrate of another, or an isotope. And obviously they're not exactly the same compounds, but they're very similar. But it is, again, more likely that it could be an error or a duplicate. And in that case, as Fiona said, get in touch with us on the help desk, and we will look into it further.
So the next question, how do I download target ligand pairs from the database? So it depends exactly what you want to do, but if you're starting from the web interface, then on the top right-hand corner, there's a download option button where you can download in various formats. As a CSV file is the one I would typically use. But equally, if you could, if you were using the API, then you could access the data that way, or if you had downloaded the whole resource, then you could access the information in that manner too. Is there anything to add to that?
No, that sounds good.
Okay. So is it possible to find possible drug targets for a newly characterized protein whose structure is not yet resolved?
So shall I
yes,
start?
On you go.
I'll have to go. So this one may be a bit tricky. So I'm assuming this means compounds for a newly characterized protein without a structure. So in that case, hopefully I'm answering your question, but in that case, I would suggest using our sequence similarity search function. So you can see whether the protein is already in the database, but if it is a new protein or a newly characterized protein, then it may not be, but you can look at similar proteins and then see whether there are any compounds active against that target. We do also have links out to structure databases such as PDB, so you can look at the cross-references and see whether there are any similarities to characterized proteins. But yeah, there are various approaches to this. But yes, please get in touch if that doesn't answer your question fully. Fiona, do you have anything to add?
No, only that it could, if those resources that Emma has just mentioned have been exhausted, it might be worth looking into ChEMBL, simply because the data in ChEMBL is available publicly sooner than anything that's within the published literature, which would then end up in ChEMBL. So if there was a new compound and there was a new target, then possibly that information might be there. Right, shall we move on to the next one?
Sounds good.
Is it possible to combine two phytochemical compounds and create a mingled structure? If so, how? I'm not sure I can answer that off the top of my head. It might be better to email us on the help desk and we would be able to refer you to a different resource. Yeah, Emma, do you have any comments?
So I'm not sure if I fully understand the question, but if it's a question of generating a structure and then searching the database with that, or a combined hybrid structure, then it is possible to do that by going to the structure search tool and then just creating or drawing a structure of your choice and then looking for similar compounds. Again, though, please get in touch with us on the help desk and we'll try and answer your question or point you in the direction of other resources.
Okay, moving on to the next question. Can I use your database for anticipated drug effects in vitro experiments? So yes, we have a lot of data on functional assays and in vitro assays. So you could search the database for relevant information. I think I would need a little bit more information in your question than what's presented there to know what exactly you want to search for, whether you are starting with a compound and you want to find similar drugs, similar compounds and their targets in vitro experiments, or whether you're starting with a target, a protein target in the in vitro experiment. Emma, would you like to comment on that?
So again, you can search for this information in ChEMBL, but it will depend on your question, as Fiona said. But yes, you can look for data that may help you to predict some drug effects. So if you want to know if a target binds to a particular protein, then of course you can look at the protein within the database and look at compounds or similar compounds that do target that and bind with high affinity. And that might help you to get an idea of what would happen in vitro experiments. But yeah, please get in touch with us if you have further questions.
Right. If I have a list of candidate compounds, can I upload all of them together and check for shared target pathway or proteins? So yes, a new function that we have recently implemented is to be able to upload a list of compounds within, I think, the advanced search bar. You may have to have them comma separated or something like that. And then you should be able to check for all of them. There is a limit on how much can be displayed on the web interface, because if you have too many targets and too many compounds, they don't fit on the screen. So you may have to chunk them up depending on how long your list is. But yes, you can certainly do that. If you need more specific instructions, then please get back to us on the ChEMBL help and we can share an example of how to do that. Emma, do you have anything to add to that?
No, that's fine. Yes, as you said, the new kind of list search option is a good way of approaching that.
Okay, the next question. Is it possible, have I answered this? Is it possible to find drug targets for a newly characterized protein whose structure is not yet resolved? I'm sorry, we've answered that already. Next one down. Following up on the natural products question, how would I filter for natural products? So if you go to the compounds bubble from the main ChEMBL homepage, go to the compounds page, and then on the left-hand side, there's various different filter options. Under one of those, under the compound type, then there should be something for natural products. Emma, do you have anything to add?
So I'll just add that alternatively, if you do have the database downloaded, again, you can use that as a filter option. So we do have natural products flagged, so you can perform various filtering operations using the downloaded database.
Okay, and the next question, what is the state of extraction of bioactivity data from ChEMBL and relation these data to corresponding compounds? So I'm not quite sure exactly what's meant by that, but we do extract, if ChEMBL is able to annotate bioactivity information for compounds and their targets, then we do extract some of those into ChEMBL, notably those for underrepresented targets as part of this Illuminating the Druggable Genome project. Emma, do you have anything to add to that?
No, that sounds good.
The next question, are compounds linked to PDB structures they are present in? Emma, would you like to answer?
So that's something I will have to double check. But yes, I believe that the cross references at the bottom of the compound report card do reflect the compound itself, but I will have to double check that. So yes.
Yeah, there's definitely cross references at the bottom of the compound report card that cross reference to PDB. But yeah, okay. So we're still going with more questions. That's great. Lots of interest. So is it possible to group and download all antiviral small molecules approved or targets?
Do you want me to take this one?
Yes, on you go.
Yes, it's absolutely possible. And there are various ways of doing this. So if you have, from the targets view, you can use the left-hand filter panel to filter specifically for viral targets. You can then filter again to specify single proteins, multi-protein targets, etc. Another way of doing this is from the main ChEMBL homepage. Scroll through the visualizations, and then you have a classification tree. So you can then scroll on that to viruses and then view all of the viral data from that approach. So yes, you can definitely do that. And then as Fiona mentioned earlier, you can use the icons on the interface to download your data as a CSV or TSV file.
Right, next question. Can we access the single compound with multiple activities, e.g. anti-cancer, antibacterial, antiviral from ChEMBL? So the antiviral bit we've just covered in the previous question.
Do you want me to take that, Fiona?
Yes, on you go.
So yes, so we have any extracted data for a compound from our data sources will be assigned to that compound. So you can download that to the database. So for example, if you have a compound report card for, let's say, aspirin, you can view all the bioactivity data that we have for aspirin, whether that's antibacterial activity, antiviral activity, anti-cancer activity, any of the activities that you have, that can be downloaded from the database, as discussed earlier, as a CSV or TSV file. And the same applies to other compounds or groups of compounds. You can extract all of the bioactivity data for those compounds from the database.
So how can I find the most suitable assay for a given set of compounds? So I think, one option you could, if you've got your set of compounds, you could look and see what assays they've been tested in within ChEMBL. That's a slightly subjective question because I'm not sure what you mean by most suitable. It depends on the purpose that you're looking for. So Emma, do you have anything to add to that?
So again, it does depend on your question, but so if you were interested in, if you had a set of compounds, you're interested in cytotoxicity assays, then you can filter within the assays for keywords such as cytotoxicity, or you could again take those assays and then filter against targets. It may be a cell line target that you're interested in, or a virus. So there are ways of filtering the assays for a given set of compounds. But yeah, we can help you in more detail if you get in touch with us on the help desk.
Okay. The next question, what criteria are used to identify natural products among compounds?
So we do, we do have a set of criteria, and I will have to get back to you with the precise rules, but yeah, there are rules that that we use based on whether they're found kind of in plants, etc. But I can't remember those off the top of my head, I'm afraid, but I can get back to you with those. Fiona, do you remember anything or have any more detail to add?
No, I would need to go and check that as well.
Yeah.
They're assigned when that compound is first included within the ChEMBL database as part of the initial manual curation.
Yeah.
But no, I don't know. So may you introduce a book for learning this as introductory? I mean, a book would be great, but I think for now we keep things up to date more frequently with webinars, and equally, if you've got specific questions, ask the ChEMBL help or look at the information that's available on our web interface. There's quite a lot of background information and FAQs that's available there. Emma, do you have anything to add?
No, other than we have the webinars, etc. available.
Where can I find the confidence level of the assay present in previous versions of ChEMBL? I can suggest one answer. Emma, did you have anything in particular to say?
My suggestion would be to use the archive versions of the previous databases. You would have to download that. So the old confidence scores won't be available in the current release shown on the interface. So you would have to go to the download section. If you're interested in the confidence scores from version 25, you could download that and then extract those confidence scores. But unfortunately, you can't get the old data through the interface at the moment.
I think if you look at the FTP site where the downloads are for the old versions of the database, there is one text file that provides the whole database schema and a definition of each of the fields, and I think it might be in there. I would need to check, but that might be the simplest place to find that. Right, the next question: how can I contribute to ChEMBL for data that is annotated in other databases but not in ChEMBL? So if you have data to deposit, then we would be delighted to have a discussion with you about that. It's the process for depositing data is a fairly new process. So if you and we're starting to move that forward, but there is now a process for doing that. So please get in contact with us if you have information to contribute. If there is specific data that's annotated in other databases but not in ChEMBL, maybe get in contact with us through the help desk because it may be that we have something in the pipeline or it may be that the way that we annotate information is just it's not better or worse, but it's just different to how another database might annotate similar information. Do you have anything to add to that, Emma?
No, I'd agree. Please get in touch with us on the help desk. And if there is any additional information that we can include, that would be great. We do welcome any feedback and suggestions.
Okay, the next question is for any additional follow-up questions, where would we write to you? So it's ChEMBL-help@ebi.ac.uk. It's written within the slides. I think the third last slide where we have useful information. And I think AJ might be replying directly to that question as well within the chat function.
Yes, I just wanted to say that I'll send the link to everyone, so they will find all the details within the slides as well, so they can find the email for help desk within that. Okay,
a couple more questions now because we're nearing the full hour. Is it possible to look for targets of a specific protein in a particular disease?
Do you want me to take that?
Yes. Emma, do you want to take it?
So I would suggest in this particular case, you can start by looking at from the indication view. So you can look at a particular indication that you're interested in, and then you can look within that indication, you can then look for targets that are relevant, and then for compounds. So similar to the dengue example, you can then start from an indications starting point. If you're interested in a particular protein, again, it may be worth starting from the target view and then filtering to look for, filtering to look at your indications. Or if you're interested in a mechanism of action, so an efficacy target, we do have drug mechanisms curated. And again, you can see other related data related to a particular efficacy target in a disease. So hopefully that starts to answer your question. Fiona?
And that sounds quite comprehensive. I would use a similar approach, start with the indication and work from there. So the next question, I have 3 different fluorotannin compounds from 3 seaweeds. All showed great antibacterial activity, and docking score with bacterial OMPs is also high. Can I find similar compounds in the database as references? So yes, maybe the place to start with this is the advanced search function. If you put in the compound, then you can look for chemically similar compounds as a particular percentage or substructures that are similar to the compounds that you have, and then look at what activity is available for those and what targets they interact with. Emma, do you have anything to add to that?
No, the only thing I would say is that in addition to similarity searches, you can also perform substructure searches through the interface. So they're both approaches that may be useful when looking for compounds similar to yours.
So the next question, how can I download toxic compounds? So interestingly, we've just in the most recent release annotated boxed warnings for drugs. So that may be one source of information. If you go to the drugs page, then you can look for drugs which have been approved but then withdrawn from the market, or drugs which are on the market but have a box warning. That means that they have a severe or life-threatening side effect. In terms of toxic compounds, that's possibly slightly more difficult to find. One thing that we do have available is we know that particular substructure parts of a compound can cause toxicity, and that's available to access. Emma, do you have anything to add to that?
So the only thing I would add is that you can look at the, as Fiona mentioned, you can look at the structural alerts. So we've curated those features. If you're interested in a particular type of toxicity, for example, cytotoxicity, then it may be that you want to start from the assays viewing ChEMBL. But if you have any other questions, or you're interested in, say, for example, carcinogenicity, you again can start from the assays point of view, but it depends on with what the particular question is. But yes, there are various ways to explore the data. But yeah, hopefully that helps as a starting point.
Okay, there's a question, will the webinar be available? So yes, EMBL-EBI will make this webinar available after the session. The next question, can I search for CAS numbers? So yes, there are CAS numbers. The simplest place to search for that, I think, might be through Unichem, which, as you remember, maps between equivalent compound structures and their chemical identifiers. We don't formally publish CAS numbers within ChEMBL because there's a limit to how many CAS numbers there are, and we have more compounds than there are CAS numbers. Do you have anything to add to that, Emma?
Just that, so no, there are restrictions on use of CAS numbers. So there may be other ways to link out to other databases, but no, we don't assign CAS numbers for our compounds due to licensing issues.
So the next question, what is the criteria for selecting patent data from binding DB? I think I would need, there are criteria, I think I would need to go and check that. So if you would like to know, then it may be better to email the ChEMBL help desk. Emma, do you remember off the top of your head?
I would pass that question on to a colleague who would know, who would have a more expert view on that.
Okay, and the last question, since we're now on the hour, I think we've already answered actually. What criteria are used to identify natural products among compounds? So we discussed this earlier. Yeah.
So just to get back to you on that question, I know we have already talked about it, but things that have come up are things like lactam, steroids, tetracyclines, monosaccharides, folate, statins, various nucleosides, nucleotides may all be classified as natural products, but we do have a more comprehensive list. So if you're interested in more detail, then please get in touch with us.
So thank you very much. It's great to see so much interest in ChEMBL, and I hope that it's useful in your research interest. Thank you.
Thank you very much.